Now that you've made your predictions, let's look at those predictions and see what the actual results of the experiment were. Saline solution is a solution that is isotonic to cells. So when you inject a mouse with saline solution, you expect that nothing will happen and the mouse will go about living as normal. We already know that the rough strain is harmless. So the immune system will attack the rough strain when you inject it, and the mouse will be just fine. It will live. Smooth strain, we also found out, is very harmful and it's deadly. It evades the immune system. So when you inject a mouse with the smooth strain, that mouse will die. The next one is a little more complicated. So we take the smooth strain and we heat it up. And when we heat it up, that kills the bacteria. So these bacteria are now dead, and we can inject that into the mouse. And because there are no living bacteria which cause the infection, the mouse will live. The last experiment is a little more complicated. The rough strain bacteria were harmless. They did not cause the mice to die. The heat-killed smooth strain were also harmless, so they did not cause the mouse to die. When you mix those two together in an equal solution, half each, since neither of the components in the solution are harmful to the mouse, you would expect that this mouse would live. If you did not get those results, if your predictions differed from mine and you don't understand why these should be your predictions, pause the video now and call the teacher over to explain it a little bit more. These are the correct answers for the predictions. If you did not have these, you should change your answers to these if you understand what we went over. Now on to the results of the experiment. It turns out that when you inject a mouse with saline solution, it will live. No surprise there. When you inject the rough strain into the mouse will also live. Again, no surprise. The smooth strain will kill the mouse will die. When you destroy that smooth strain, kill it, the mice will live. And then this last one, when you take the rough strain and the heat-killed smooth strain, which are both harmless, and you inject them together into the mouse along with that saline solution, all three components in that syringe are completely harmless, yet when you inject them all together in combination with each other, the mouse dies. Continue with this activity to find out a little bit more about why that happens.